it's a beautiful auditorium. I had the chance to tour this room just a year or two after it was built, and they showed off all of its amazing capabilities. And so it's a delight to have the opportunity to speak here, to come back to Louisiana, to see how this meeting has grown. Am I correct, Gus? Or I believe I spoke at this meeting in its first or second year when it was getting going many years ago, but I think it was up north. Shreveport, maybe? Not positive. In any case, I will try and keep us on schedule. I set a timer over there. So you've probably already learned I can talk really fast, okay? So if I begin speaking too quickly, just wave the hand, right, and slow me down because I can get pretty excited about the work that I'm working on. So I've been giving talks for the last couple of years on this theme about unleashing your inner data scientist, because I think at our core, most of us really want to mine all of that data that's out there. We know that that answer to our research question is probably sitting in some resource somewhere if we could just get to it, if we could just use it, right? And so whether you have to learn to purify a protein or write a little script, we're all doing science and we're trying to get at where we're going. Now, speaking of getting where we're going, I had to get here today, all right? Now, this is not a shot of my purchasing my tickets to come here. It was actually, I think, about Christmas time I was going to go out and visit my sister in Seattle. But it's one of these scenarios that has intrigued me for years, that I think most of us have done this. You can go on the internet and you can search for flights from competing companies from all over the world, right? You can filter those results, right? Which one's here, right? You want a non-stop flight, single stop, double stop? What time do you want to take off? What time do you want to land? How much time do you want between your connections, right? Really, really quickly, from companies, right? How many seats are left? How much is it going to cost you? You can sort them and filter them by the price, by the connection time, by whether or not you have to spend the night in the airport, right? And then what do? They are kind enough to forward on your information for you to the car rental company, the hotel company, all to make it easier for you to spend your money, right? And accomplish something. So you kind of have to ask yourself then, why can't we do this with our biological data, okay? Because the technology is there. And I spend a lot of time thinking about this. And I think there's a number of reasons, and I would love to hear what people have to do, right? And so how do we ask biological questions involving data located elsewhere, right? Because that was what the search engine did. It brought together data that were located elsewhere, right? And made it easy, visually appealing, and it performs a function and a service for you that you benefit from, so you like to use it, right? And so if you think about this, right, many repositories exist, right? Who here has gone to a data archive? GenBank, yeah, right? Unipro, great number, PubChem, PubMed, right? A lot of databases, right? And so how do you access them, right? Your browser, right? Many of us have to do that, right? We're still having to use a graphical user interface to interact with each repository, right? So when you go to get your airline tickets, you don't go to American and then United and then Turkish Air and then Canadian Air and South African Air, all right? But that's what we do in science, okay? So this is it's an interesting conundrum as to why we do this, right? Now you can set up some API or some web services that could do some of these interactions for you, and more databases are making themselves accessible so that you can do that, all right? And then, but even when you go there, we're pretty limited to querying for a keyword. And actually the same was true when we were doing our flights, right? Only airports are allowed. Right, it's a very controlled vocabulary, right? Only certain cities, only certain specifications, certain calendars, right? Everything is very, very controlled, right? But it's still frustrating when you have a lot of things to do. You're working on a large omics project of some sort. You're doing some large screen for whatever your favorite organism is. I'm going to talk about infectious diseases today, but what I'm going to talk about is relevant to anything. So just insert your favorite research paradigm, okay? And so then you go to these research repositories one at a time. Maybe you can put in a whole FASTA file of sequences or a list of gene IDs, right? But then, what challenges do you encounter when you try and get data from these different repositories, right? It's frustrating, right? So, they're all together, sorry about that, right? First off, you probably discovered that some data sets were mapped to a different genome version, and all of a sudden the IDs are wrong, right? That happens a lot, right? Sorry, your data might be out of date. So you find that, well, yeah, they did predict gene ontology terms or PFAM domains or whatever back in 1989, right? Whenever you got your data set from, right? And then you have to ask yourself, how much time do people in the lab, postdocs, grad students, undergrad, is spent just trying to take the data that you got from these different repositories and put it all together in a form that you can use because you really have a more interesting biological question to ask, okay? And so that's going to be the thrust of my talk today. And so part of this is being caused by this explosive growth in sequencing data, all right? And I'm going to date myself. I graduated college right there, okay? And GenBank used to arrive on tapes, right, for years. And you could get your data, and that lasted all through grad school, right, when you still thought there was a lot of data, right? And the problem of data and managing data has become huge. And it's no different than we've had to develop robots to deal with plating out things into 384 well plates. Right? Well, we have the same issue when we're trying to deal with very large data sets, okay? So I'm not going to belabor that. But what we did think back then, right, there's a number of things. Oh, if we just had the genome, if we just had the genome, if we just had the genome. So a number of genome consortiums, a bunch of GWAS studies, oh, we're going to do 1,000 genomes, right? Now we want to look at 1 million people, right, for personalized medicine. So I was part of this. I've been involved in malaria research for a long time. It's caused by an organism called Plasmodium, transmitted via mosquito bites, right? Still a very, very significant pathogen that causes a very, very large number of deaths, primarily in children under the age of 5, okay? And so here we were, this was quite a while ago. We had the parasite genome, we had the human genome, we had the mosquito genome, right? So surely the answer would emerge, okay? But it didn't. In fact, we discovered we needed even more data, right? As most of us often did, all right? And so what's the biological equivalent of a travel search engine for a biological system, all right? You might want to find all genes that maybe you're studying something that's located on a particular chromosome, near the ends of chromosomes, subtelomeric regions. Maybe that encode a signal protein because you want something that's targeted or secreted. Maybe that encodes some kind of motif, and I forgot to finish typing my motif there, but CCXXY followed by 15 of anything, and then a basic residue, right? Something that you happen to find in whatever group of genes that you're studying, all right? Maybe for those genes that you find in step 1, you want to know which of them have evidence of expression because you want to know that they're actually used. When are they used? What time are they turned on? At some developmental stage in a particular tissue. And then you probably want to know maybe, well, you're interested in something that's signaling molecule, so maybe it has to be phosphorylated. So you want to look at phosphoproteome data, right? And then maybe you want to know, hey, the immune system sees it, right? And so you want to know that it's under diversifying selection. And to do that, you need population studies. These are the kinds of things we dream about when you're sitting in a restaurant or a bar or lab meeting and you're talking about if we could just find, right, the X or the Y or the Z that has the characteristics that fulfill the criteria for whatever the scientific problem is that you have at hand. All right, and so why is it so hard? All right, and I've already told you, the technology is not the reason why this is so hard. The technology exists. We can do it for airline companies, we can do it for car rental companies, we can do it for hotels, we can do it for colleges, you can do it for any number of things, right? The technology exists, all right? But there are differences. There are very real reasons why this just hasn't happened. The first is the subject matter is different. Our subject matter is much more complex. Maybe you need 1,000 terms to handle car rentals and airlines and aisle seat, window seat, middle seat, Is it are you in the comfort section or are you back in the bus? Where are the bathrooms on this model plane? There's a number of vocabulary terms and they're fairly well defined and not too many people will argue about what constitutes a window seat versus what constitutes an aisle seat. The same is not true in biology. People will fight you to the death over whether or not that's blue, okay? And the incentives are different. All right, with travel, there is money to be made via cooperation, all right? The corporations are global and scientific funding is not, okay? And so the challenge is to come together, to agree on vocabularies, to build these kind of overarching schemes that can link together the different national and or international data repositories are exceedingly complex. And then you add to that the wonderful little panache of academics, and that is we all like to create something new and something special, and of course it's better than what anybody else has, right? And for a certain extent, we're rewarded for that. That's how our educational system and our research system works, all right? But it's counterproductive to getting some things done in terms of being able to share data. All right, so with that as kind of a backdrop, I work in the infectious disease world. In the infectious disease world, we have model organisms. There's a tremendous amount of data about humans and where they live and the climate. There's surveillance of this climate, of spreads of diseases. There's pathogens. There's a whole medical informatics world with electronic medical records and/or trends in epidemiology, vectors of disease, whatever they may be, and the disease biology itself, whether studied in human hosts or in the model organisms that we use to study them. And so there's a tremendous amount of data coming from a very diverse set of communities. Okay? And these data might look something like this. You've seen genome annotations, some BLAST results, your expression profiling, some SNPs, some synteny, some phylogenetics, some isolates collected, maybe a cellular phenotype, a protein structure, protein localization, your protein expression, your orthology analysis, your RNA-seq, your Go terminologies, your protein-protein interactions, your ChIP-seq, your metabolic pathways, maybe some interactions, the immune response, your hosts flowing around, the climate change, and making some models. That's just a few. And you're supposed to put it all together and answer the question, okay? It's hard, right? That's what we're trying to deal with. And it leads to this, all right? And so I think manual data processing, which is something that we spend an incredible amount of time doing right now. If we were to count up the total number of research hours spent just trying to get, retrieve, and use data sets, it is an incredible amount of productivity that is quite lost, all right? And so it's really a very serious problem in the data analytics world for helping the medical community and the research community to get along. So can we imagine a world in which the data are easily accessible, in which everything's been digitized, there's some kind of format, we know how and where to find them, and I really look forward to the world in which data are self-describing and easily discoverable, where databases can talk to each other, and where file conversions are not needed. 90%, this has been quoted like all over the place, and Bioinformatics is just converting things back and forth between whatever file format you need to get them into the next program, okay? And so, we as biologists, or we as data scientists, wherever you find yourself on the spectrum, right? Somewhere we have to converge and we have to meet, in order to make some of this become a reality, all right? We won't have everything talking to everything, but we can do a lot better than we are right now. So, to that end, and here I'm going to let my University of Chicago roots shine through because they're very into Frank Lloyd Wright, and he really was a proponent of organic architecture and basically that form and function are tied together. And I think this is never more true than when you're dealing with data. And so I don't know how many folks in the room regularly build databases, but the way in which you decide to store your data, whether you're going to go with a relational structure or not, if you are going with a relational structure, how you design your schema absolutely influences what you can do with those data after you've put them in that format. All right? There are some things that you'll be precluding based on an architecture that you will choose, or that you will be enabling based on an architecture that you choose. And so a lot of thought has to go into how to do this in order to maximize the utility and to know the target audience that you're actually going after using the data sets. And it may be that data sets need to be in multiple formats to facilitate multiple different types of activities. All right? Making the tools is not easy, all right? Especially making tools that really serve a function and address a need, right? I never do anything that there's not a clear use case and an absolute demand for. We can build lots of things, right? But not everything is useful just because you can build it, all right? And getting meaning from data, right? And that's not the same as truth, right? It's really difficult because data are contextual and some of them are actually hard to get at. So for example, you can know that there are data about users of a bank, but you can't get into their records and see what else is going on, right? And the same is true for a lot of human records, a lot of clinical records, a lot of data that are not meant to be accessible under certain circumstances, all right? The other really important thing that I find gets often forgotten, especially when we're drowning in data, And that is, when you don't find something, it doesn't mean it's not there, and it doesn't even mean it doesn't exist. If people haven't shared it or they haven't generated it, you won't find it. And so this is really a key problem. Some folks will go and search a database and, it's not there, all right? And it could be a faulty search algorithm, or it can be that there's a whole section of taxonomy that hasn't been explored, or of protein modifications that haven't been cataloged yet. And so there's a real, issue with declaring something to actually truly be a true negative, right? Or to say that a gene doesn't exist in a genome. This is the most common one that happens. You search a genome annotation, you don't find it, all right? And you think that your gene isn't there, but it is there if you were to go and search for it at the sequence level, or perhaps the sequence assembly isn't complete. Most genome sequences are not complete. Therefore, it wasn't generated. You might be missing that region of the chromosome. If you're missing that region of the chromosome, if that's where your gene happens to be located, you don't know for sure that it's not there. So other things have to be done, all right? And the final point that I want to make, and I'm going to harp on this a lot for the rest of my talk, is that any piece of data is nearly useless without its associated metadata, right? As an example, let's say you go do a blast search, right? You take your sequence, and you want to know what it's closely related to, and you go, hey, we found a great hit. Here's this other sequence, right? Your E value is, 10 to the minus 69, and these 700 nucleotides are perfectly aligned. But we don't know what it is, right? They don't tell you what organism it came from. Or they don't tell you, if they tell you the organism that it came from, you don't know where on the globe that organism was sampled. What population did it come from? There's extremely rich metadata that make interpreting your data highly relevant, right? But it's difficult if you don't know that metadata. And increasingly, for those of you who've had the pleasure of writing grants recently, I'm a big fan of the efforts to capture more metadata, to make data public, to make data shareable. I'm not a big fan of the way it's happening. I don't think that just dumping them in repositories makes them shareable, and I'll show you that in a minute. All right, but we have to begin this process of collecting data about our data, because that is what really makes them useful. Okay, so elements of a successful architecture, in my mind, are tools built by biologists for biologists and clinicians and epidemiologists and modelers, and they will provide the ability to mine, to visualize, to search, to compile, and to visualize and explore the data dynamically, all right? Not Google, type a word in a box, see a catalog of everything, but to actually generate hypotheses based upon data that you find, all right? So towards that end, I'm going to tell you about some databases that we work on where we're trying to realize this vision, all right? Where we want to take data on organisms and their pathogens. We want to look at genomics, every kind of omics that you have, including clinical epidemiology data. I'll show you in the end. We want to mine it. We want to visualize it. We want you to play with it. We want you to be able to generate and test hypotheses based on the data, not just go and download it, okay? It's a very different type of mental mindset. So the databases I'm going to tell you about are called UPathDB. It is an NIH-funded Bioinformatics resource center. It's one of 4. We've been in business for about 20 years now. We grew from one organism, the first organism, which was Plasmodium and PlasmoDB. We're up to 289 organisms, and we cover a very large number of organisms now, including host, because host is the only thing that all these pathogens have in common. And normally people who work in the parasite community don't talk with those who are in the bacteria community, who are in the viral community, but these pathogens are very often invading and then evading the host, probably utilizing some similar strategies, but you wouldn't discover that if you couldn't walk across them. Okay, these organisms, no need to bother or remember any names. The only thing I want to point out is humans, fish, fungi, and amoeba make up this part of the tree, all right? There's plants down here, and the rest are mostly protists, and they're very, very diverse, okay? They're really different from each other. So when we're over here in the excavates, or we're over here in the amoebas and the fungi, or over here in the apicomplexan organisms. We have a database for each of these major groups. Every community has its own color and its own branding, but it's the same database under the engine. All right, so what kind of data types do we have? Well, they're endless. We just keep adding more and more, but we handle all the genome sequences and their annotation, orthology predictions among all of the predicted proteins, synteny predictions, all the transcriptomics and protein profiling, field and clinical isolates, including with their metadata, chromatin modification, ChIP-ChIP, ChIP-seq, functional assignments of genes, phenotypes if they're knocked out, is it lethal, is it not, what do you see? Metabolomics, polymorphisms of population genetics, a lot of that comes from resequencing, the literature, and much more. What we do not do is curate. We don't sit there and read papers and go in and fix things. The community can read the papers and go in and fix things, and they can link their papers to this resource. Our job is behind the scenes to take all this data and solve those hard problems of linking them together for the community. All right, so this database is called PlasmoDB. I realize that much of this may not be overtly readable, and that's really okay, all right, because everything that's important will get blown up. So at PlasmoDB, I'm going to take you through a sample search. You'll notice first the page doesn't look like just one box where you go put in a word. All of these are different types of searches that you can conduct to find genes, other data types, or some tools. We're going to go in and we're going to do a search and we're going to look for a protein, and we're going to look for proteins in this whole genus of parasites that have predicted signal peptides. So we do that by selecting it. I'm going to take all of the organisms, I really don't care which one, and I end up with an answer, right? So there was like, what, 106,000, right, genes have a signal peptide. I spent 2 years of doing human-computer interaction studies to learn that a red add-a-step button is irresistible to a biologist. Okay, tried all kinds of other things, didn't work. All right, so signal peptide and what? Okay, so the signal peptide results are shown down here below, and you can do a number of things with them that I'll show you in a minute. But what I really want you to do is hit that red add-a-step button. So if you do it, we can then add some more things. So now we're looking for a new search for genes that have expression. I want to look at RNA-seq data. I'm going to pick this blood stage, so I want this expressed when it's in the human host in a red blood cell, all right? And I can check that I want it expressed at 8 hours, okay? So you pick that, and then the really cool thing happens down here, all right? And that is we sneak in Boolean operations under the guise of Venn diagrams, all right? And then we get users to say, well, how do you want to relate this search to the search you just ran on signal peptides? I want to intersect them. I want them to have a signal peptide and to be expressed at 8 hours, okay? So if you do that, we begin drawing a strategy of your logic for you and showing you the results of each step. And you can go and modify it, and you can make really elaborate branch structures, okay? And you can save them, and you can do all kinds of things with them. But we can add another step, right? It's a lot of fun. It's addictive. Now I'm going to look for orthology. I took any species in the beginning, but now I need to have those genes that are only orthologous to a particular species because that's what they're working. And so in this case, gone and picked Plasmodium vivax. It doesn't have a lot of expression data, so we're using other species as an inference, right, to make a transference about this. And we come up with 140 some orthologous genes. And you'll notice the shape has changed color to tell you you've done something fundamental like changing organism, okay? And now, cool things can happen. You can save this result, you can duplicate it, you can share it. We give you a little tiny URL, you can paste an email, send to your advisor, your coworkers, your collaborators. They can open it up, they can see your strategy just as you left it, all right? And they can continue to work with it. Or you can come down here and you can look at your results and you can do a sanity check, for example. We can add a column down here and say, I want to see that gene expression because I want to know that they had high gene expression at 8 hours. So we do that. Now for each of these 140 some genes in vivax, well, sure enough, even though we did the search in a different species, this is the 8 hour point mark right here, we can see that they all have very high expression at the 8 hour time point. It's kind of a sanity check. Okay. All right, so now I want to tell you a couple of other things that you can do. We've added some tools now, so we can come over here and we can analyze the results further. We can go in. We've done gene ontology enrichment tools. We have metabolic painting tools, so you can, for example, say, well, this gene set that I found, put them on a particular metabolic pathway, and then you can look at gene expression of each of the members of that pathway. You can ask about phylogenetic conservation of the genes in that pathway, any number of things. So we do the gene ontologies. Pick enrichment, we say for molecular function, search interpro, and it calculates everything for you on the fly, and you get not only your p-values, but your Benjamini and your Bioinformatics multiple hypothesis testing statistics coming out. And we now have a link that will let you take these straight to another engine called Vigo, where you can actually visualize and play with your results a lot better. So we don't try and reinvent the wheel when we don't have to. All right, so then for any particular gene that you're interested in, like any database, we have a giant records page where you can look at any of these number of data types, and for the sake of time, I'm not going to walk you through it, but you can, there's little shortcuts up on the top for any particular data type that you're looking for, so you can jump around and you can see any number of pieces of data of whatever we happen to know about this gene. Many people would come to a database to browse this kind of encyclopedic information, but you can use it as a data mining platform as well to generate genes. We have the gbrowse installed. We're moving to jbrowse. You can look at RNA expression studies. You can look at synteny plots among any number of different species to see how things are conserved or not. So, you know, the typical things you would expect in the genome database. All right. That's what we did for, you know, the first 15 years. The last few years, we've really been taking on a number of different roles. We really have turned ourselves into a metadata integration platform because we realized that let's say in the case of Plasmodium, there are now 10,000 genome sequences. Nobody goes and browses 10,000 genome sequences. What makes the 10,000 genome sequences valuable is how they differ from each other. All right, what makes them unique? And what makes them unique is being able to associate their SNPs or their copy number variation or their inversions with populations that they come from. Are they resistant? Are they here? Did they come from this part of the world or that part of the world? And that most people will now use a platform like ours to find the right data set. When there's 10,000 of them, it's much better to go in and mine through the metadata to identify the data set that you're interested in and then take it and carry it into the tools. We've created a workspace that I'll show you. We've created a private galaxy where users can now take their own data without ever giving it to us, go up into the cloud, run either RNA-seq mapping or SNP calling against our reference genomes that we're constantly releasing every 8 weeks. And then bring the data back and look at it in the browser without ever submitting it to us, right? So they can look at their data in the context of our data. And we're starting to take data for systems biology, and I'm only going to briefly mention that at the end because it's really exciting. So this is just our Galaxy Cloud platform and the number of pre-configured pathways that we've made available to the community. And the CPU time is free. You just take your data and go right up there and use it. All right. The newest thing that we've been working on, where I want to spend the second half of my time, or not quite the second half, is the ClinEpi database. So we realized that there's nothing particular about our platform to mining genome data, right? So you can ask for all genes on this chromosome and this organism that have this signal peptide that are expressed at this time. You could ask for all specimens in a museum collected from this country for this species, all right, by this investigator in this year. Or you can ask about all patients seen in this study that had this clinical outcome, right, that happened to have data set X or Y, or had something along these lines. So we just released this, came out like 6 weeks ago. I think it's February 1st, February 15th, somewhere in there. And so we are funded to do this by the Bill and Melinda Gates Foundation. They've given us their large studies on malnutrition. I shouldn't say they've given it to us. They're working with us to make their data public because they've also embraced the data sharing and they want their data to be accessible to other users. All right, so it's a resource to design to explore and mine epidemiological data and data sets. All right, and so, but before we can do this, I'm going to take a little detour. Because remember, again, I talked about why it was easier for airlines to match their data up because we all agreed what's an aisle seat and a window seat. It's not clear in the biology world. All right, so we spent a lot of time working on harmonizing data and working on ontologies. All right, and so we were part of a working group that was developing standards for human pathogen and human vector interaction type data sets, including characterizing hosts, specimens, how the specimens were processed, how the pathogens were detected, how were the pathogens isolated, how were the samples managed, how were the data transformed or normalized, how were the sequencing samples prepared, what kind of sequencing assay was done, etc., for these types of studies. And so we spent months, and we still spend, we have an entire team dedicated to ontologies, to just realizing that if you live in a dwelling, it has a geographic location, and participants live in dwellings, okay? And participants have ages, and all of the various characteristics. And so we're part of the OBO Foundry, and we work a lot with the NCBO ontology databases working on this. So once you have that in place, you can now begin taking large numbers of data sets and harmonizing them across their data dictionaries that describe a number of things. And so, like before, I'm going to show you a number of web pages, but just imagine, you were working on cancer samples or developmental stages of C. elegans or whatever it is that you happen to be working on in your research, all right? And so, like before, you could come in, it has a pretty new graphical look, but it's the same engine driving it, all right? So we can come in and we're looking for participants. what kind of participants, and it tells me immediately, I'll read this with like Bangladesh, India, Kenya, Mali, right? So what country do the participants live in? And we're showing you that distribution before you even click. And this is something that's new, because before you could say, give me all genes with a signal peptide, but we didn't tell you how many genes with signal peptides were there, right? Now you know whether or not you're going to get yourself to a null empty set, right? Well, how much you're restricting yours. So you can go in and you can select by country, all right? You can select by gender, all right? So we took from all countries, but we took only males, all right? And you can immediately see that we have cut down on the number of participants because now the half of the gray bars are there. So we have selected 130 out of 240 households, for example, okay? And then we might go in and select further by their weight relative to their age, because this is a diarrheal study, okay? And so, again, we can go in and we can pick a particular age range, all right? And then we can go in and look at the distribution that would be there for the whole population that's in the database versus what has been selected. And in this case, they did the selection based on z-scores, which is the way epidemiologists like to think about the weight per height or various physical attribute measurements, okay? And then we draw for them the same kind of strategies that you saw before. So we go in and we took, where we knew there were diarrhea samples, and we were comparing males that had a positive height for D statistic versus a negative height for Z statistic. We ended up coming out with 26 participants, okay? And again, we have the same add a step. But now, we're not looking at genes, right? We have a participant with an ID that cannot be traced back, right? That came from Quito, Peru. And now you're able to look at, this is every time, that the child was seen, all right, over a very long period, and what is happening, all right, with a number of statistics that are being captured and number of diarrheal episodes. They had 21 diarrheal episodes, right, during this time. Okay, and just like we could look at for many, many genes, you can look at this for each of the participants, all right? And again, it's kind of a sanity check about what it is that you tested for. But you can test for any number of things. So we went and added in show me gene plots, right? But we can look at whether or not there was alcohol intake during pregnancy, what was the maternal age at the birth of the child, was there the mother's marital status, right? Any number of things that you can add that exist in the data set to go in and profile further. All right, this is blown up really big now, so you can see this is the height for age, weight for height, and weight for age. Z-scores plotted out all the way over the time, all right? And so, again, where we're looking at plus or minus 2 standard deviations when there's diarrhea, and these are in the red lines, okay? All right, so now, what we've enabled here is we've imported our Shiny app tools. And so now, with the data, we can actually let people begin to explore it. So they can do some contingency tests, right? So you can come in and you can take your data set, that set that you've just identified, and you can go in and you can have a look at them, all right? And you can go in and you can summarize and get summary statistics about this particular population. And again, you're able to do it in a way that is not risking exposure of the participants, because the way in which we have presented the table and because these are summary statistics. So that's really kind of cool, all right? And again, you can look when you start summarizing things, and you can begin to then, you get your error bars on here, and you can plot how these individual children, all right, upper or lower, had compared relative to the background population of whatever it is that you were particularly studying. All right, so I'm going to show you I just want to walk you through a little feature now, all right? And so, like, one of the things that you can do is you can go in and ask a question like this. So GEMS was particularly, this was a case-controlled study for acute diarrheal episodes. I wanted to know, was there an association, of between Giardia compared to Cryptosporidium, these are 2 nasty GI parasites that you can get, with moderate to severe diarrhea in the 2nd year of life. So step 1, what is the relationship between Cryptosporidium and moderately severe diarrhea? And I have to tell you, I have to put the disclaimer on here about all of the statistical tests, all right? We're not claiming to replace an epidemiologist. This is meant to be hypothesis generating, and that you can go in and have gross overlooks, but then a true study with the permission of the provider, right? So you can contact the provider and get the real data and conduct more in-depth studies, right? So what is the relationship between Cryptosporidium and moderately severe diarrhea, 2nd year of life? You can go in, you can screen, you can get the 2nd year of life, so from 12 to 24 months, you can just drag it. And when you do that, you immediately reduce the number of participants down to 7,500. You can then go after some personal characteristics. We can analyze a number of results. This is kind of confusing, I'm sorry. We missed a slide in there somewhere. Let me go back. All right, so we go to so we're down to 7,500. We say we're going to view them so that we can do it. We're going to do it to the analysis. We can go into the contingency tables. And now we can say, well, we wanted to know a couple of things. Their microbiology had to be positive, and we wanted to know for those who are in crypto, all right, for variable 1, all right? And in the case control, and you compare it to that, and you get a log odds ratio of 1.9, all right? So we can go and put this in, and we can go and ask the same question about the relationship for Giardia. Same thing, microbiology positive, you have Giardia relative to your case control, and you come out with a 0.57, all right? And so clearly, there is a much higher odds ratio of cryptosporidium being associated with moderately severe diarrhea than Giardia. And again, it's hypothesis generated. He's data mining in other people's data sets that have been made available for reuse. It is a truly powerful thing to be able to do. And so I'd just like to finish up then with a couple of things that I'd like to proselytize about a little bit as I think about this. I've been working on it for a lot of years. I'm going to give you this sort of in the perspective of thinking about malaria. Malaria is a very complex disease. You know that to actually have disease as opposed to being infected is an interaction between the pathogen and the host and the environment. Right? It happens all at the same time. And because not everybody who is infected becomes sick. Not everyone who is sick is infected with that pathogen, et cetera. And it's a very, very complex interaction that leads to the clinical outcome that we would call malaria. All right? And so as I began this talk, I was again saying discrete data, like kind of airplane, day of the week you want to travel. Discrete data are pretty easy to classify. And the same is true for some biological features, right? When we can agree what is a nucleic acid, what is a protein, and what is a protein structure, chemical compounds, and we have databases for these individual data types, and we have ways to talk to them. And as I said at the beginning, we can go visit this database and that database and another database to be able to pull out these data, all right? But linking data to their metadata is really hard. In the lab, we tend to do that with barcode. In the grocery store, they do it with a barcode. And that's how they know how to recall all products from, whatever that need to be recalled, right? And so you're doing an experiment. In our case, I was involved in a systems biology project that was conducting 100-day-long malaria infections, okay, in non-human primates. And so you know the monkey, you know the species, you know how they're caged, you know their diet, you know their fevers, you know their parasitemias, you know everything about them for months. Okay, a tremendous amount of clinical data. But because we're dealing with a very, very large project and a large number of samples, every sample is barcoded, right? And blood samples that are taken from certain drug treatments at certain days and certain whatever end up having metabolomics done or immunology profiles or RNA-seq or whatever it is that you choose to do. And because we have this beautiful infrastructure, we're able to take any given sample and link it back to the animal that it came from, the conditions, the pathology at that time, what they ate the day before, which for metabolomics turns out to be really important, okay? And all of these data. And so I spend a lot of time thinking about how do we do this in a larger context, right? How can we facilitate this for a researcher at large? Because I'm gonna be honest, it's a pain in the ass, okay? And that's one of the reasons it isn't done, to capture all of those details. Like what kind of software, what kind of system would we need that could help facilitate the capturing and the logging of the data as you're doing experiments so that you don't take your valuable research time and spend 6 hours of it typing endless things into Excel spreadsheets, right, which are a great currency in the laboratory, but not the best for mining overall. So I think it's really a challenge. How do we scale it up? And I think to drive this point home a little further, I'd like to do the thought experiment. And the thought experiment is how do you submit to a repository an experiment? Okay? And I just want to repeat that again, an experiment. I differentiate that from a data set. So we all do experiments that generate data. And then, oh yeah, that sequence data, there happens to be a repository for it. Maybe you did RNA-seq, so you're going to put some reads in SRA and GEO. So hey, I did it, checkbox, all right? But the experiment was more than the RNA-seq data. You did some cross, you isolated some specimens, you treat it with a drug, you did something, you knocked out a gene, and you were trying to study the phenotype. Capturing that other information about the data
historically that role has been played by the literature, right? You would go and read the paper, and that's how you would know what people did. There's too many papers, right? And there's not a standardized vocabulary for capturing that other experiment. So we have the lofty ideal that our funders and scientists as a community are trying to go towards, which is making data reusable. But what do you have to capture? What's essential to capture? We have all these minimum data standards, Miami for microarray experiments, right? What do you have to capture to truly make your data usable by someone else, right? And I'm going to argue, in some cases, we're doing ourselves a disservice by, I drew this, imagine this is a pie, right? We're taking parts of our experiment like slices and cutting them up and sticking them in places, right? But the meaning of the experiment is the whole pie. To truly understand what's going on, you need the context. You need the other data sets. And you in the lab, to write that paper, to do the analyses, have integrated them, and you then promptly tore them apart and sent them to repositories, right? Where the next person who wants to use them has to put them back together again. And what do you need to be able to put them back together again? You know, what was the big picture of the experiment? What were you trying to do? Why did you set it up that way? What were you trying to prove? What kind of patient or animal or host were you using? So many things don't even say what they're from, and it's just kind of assumed, I think, that everybody works on human, but not quite the case. What analytical procedures did you use? The kits, the machines, the extractions, they all cause biases of different sorts in the data. So to be able to capture that, right, it's really important. The actual results themselves, you know, we have a lot of data. Like I did this, you know, we generated 35 terabytes of data in this experiment that we did, okay, for the Center for Systems Biology. And most of the data didn't have a place to go. The largest amount of data was nearly 9 terabytes, and it was telemetry. Where do you deposit telemetry? Where do I deposit monkey fevers, monkey parasitemias, monkey weights, stool samples, right? The data that were needed to have this global picture of what was going on, the repositories just don't exist, all right? And the description of the experiment, the time series, what does it mean that this was time point 1 and time point 2 and time point 3? What happened? How does that relate to the drugging schedule or the infection schedule or whatever it was that you were doing in your experiment? These types of data are missing from data sets that we deposit, all right? And they make it very, very hard for the community to actually use the data in an effective way. And especially if you're doing experiments year after year and they have some kind of relationship to each other, it's really nice to capture that, to know that this is phase 1, phase 2, phase 3 of a longer series. Okay, so I want to finish up. We're taking data from this experiment that I've just described, which was called MAPIC. It's the perfect kind of case study in that the non-human primates, and here we can really play with the data because they're not human subjects, so their clinical data can be made public, okay? To explore linking clinical epidemiology studies with all the molecular omics studies with a host pathogen interaction. And we've also created MicrobiomeDB, but for lack of time, I'm not going to discuss it. And we have all of these samples from these animals. We have all of this data. What would it look like to begin to try and pull all of this together and actually have a resource where you can query when you're seeing this type of clinical phenotype, what is happening at the molecular level or at the microbiome level together, if you knew how the glue put all of this together. Okay, so I've spent a lot of time talking about databases, many of which I'm going to imagine you've probably never heard of before today. Okay. If you work on infectious diseases, these are very, very highly used resources. And so as of the end of last year, we are maxing out, if you add up all of the database total, we're almost 95,000 users per month, unique users coming to the databases. So these are really heavily used. To be a user, you have to actually come more than once a month, same IP address, you have to stay for a while, you know, we exclude all the robots and stuff like that, right? And we're approaching 15,000 cumulative literature citations at this point as well. So these are very, very heavily used resources. And I'm not standing here telling you that to toot our own horn. Databases are in trouble, all right? So at a time when everyone, you know, big data and data analytics are the buzzwords, right? The cost efficacy, how hard it is to conduct this work, the time that it takes to integrate these data, to curate the data, to annotate the data, are taking so long that repositories are being closed. The NeurOspor database was shut down, the Aspergillus database was shut down, okay? And a number of other databases live in fear of being shut down. And there are other databases that are very heavily used and in which NIH has invested large amounts of money to bring together and to curate data. We need these data. I'm an advocate for data. You can probably tell I'm passionate about data, right? But it's a scary time, all right? It really is a scary time for us right now. And so you should be an advocate for your local database. Okay, so you may recall I began my talk with our great hope of, we had the parasite genome, we had the human genome, we had a mosquito genome, of course we will come up with a cure for malaria. And we know that it's really, really much more challenging than that. And this is a bioinformatics symposium. You are bioinformaticists. I encourage you to just take that walk in the data and build those tools that are going to link things together, that are really going to allow profound insights to emerge and to generate hypotheses that allow us to spend our research dollars in very wise ways. I have to thank the very large team of people that are involved in this project. We are nearly 40. We're located on 3 continents. We get together every 18 months for a face-to-face retreat. This was the last one taken at Philadelphia. Our funders for helping to make this work possible. And then I think, as always, our goal is to enable users in the lab to generate hypotheses and to make further sense of the data. And I will stop there and take questions. Thank you.
So, I guess I'm getting kind of conflicting impressions of what you're doing here. Is this a centralized database that you're doing, or is it like a spider, or?
It is a centralized database. It's a little bit distributed, but it's under our control.
Okay, so the question I have then is, you talk about the fact that we've run into this problem to where just this whole fact of integration that's made problematic by the fact that people, for instance, when they design databases, they have things like different surrogate keys, for instance, different sessions that they use to refer to the same thing. I suppose by having a centralized database, my question is, how does that not inflate the problem?
It doesn't. We've definitely built resources to serve our community, and in that sense, we've sliced off. What we have done is, as a rule, we've set up quite a few rules. So, for example, when an archival database exists for a data type, we accept the data from that resource. So we help the researcher to put their data into that resource so that we can pull it from there, okay, as a way of sort of guaranteeing some uniformity. We have joined many other communities together to come up with developing the standards and the nomenclature. We don't want to reinvent the wheel. There are thousands of ontologies out there. We have adopted basic formal ontology as our logical structure for our ontologies, but to the extent that we can use other ontologies that already exist, we will only create a new word with the blessing of the ontology consortium to put that in there. And so we're not creating our own vocabulary in isolation, okay? What we have done is we can't accept everything, right? And I think Lincoln Stein used to refer to this as kind of the vacuum cleaner approach, right? I mean, at a certain point, there's only so much that you can take in, which is why I use the airline analogy. They're not in a centralized repository. They are for their own airlines, but they have agreed upon rules to communicate with each other, right? And the community at different times over the last 15 years has tried to come together and agree on these standards, and the technology keeps changing to allow that to happen. But I know I'm an advocate for a distributed model. I think different organisms and different communities need their own specialized resources. We just want those resources to be accessible, ideally in an automated way, in a controlled vocabulary that allows people to ask their own dynamic questions of what they need from those data. Does that help?
Yes, it does. Thank you.
Okay.
Yeah, go ahead.
So I might have missed this part. Do you have any database that would integrate structural information on protein structures and connecting it to genome variations and other things to be studied?
Not that I am aware of. I know that we have, when a PDB structure exists, for one of our proteins, we have that information, and we can also tell you if there are SNPs. you can then find those proteins that have SNPs for some number of things, but you then have to go to a third-party software that allows you to map those on to the structure. And I'm not aware of any software that by itself can make a de novo prediction of the consequence of that change on the protein structure. Usually that's when the specialist has to get involved in what they know of the protein interactions. But yeah, it would be really useful. Because now we have so much SNP data, and we know there are different consequences, right? And so having an easy way to get those mapped onto, that's one of those beautiful use cases, right, of a community-driven, really great idea, right, of something that needs to be made. Yeah, good question.
Great.
Yeah, Mike.
Yeah, early in your talk, you mentioned that the UCAT-DB suite is one of 4 cores of the NIH funds. What are the other 3? And is there communication between the cores to help learn from each other?
Ooh, great question. So yes, there are 4, and we are divided up along phylogenetic taxonomic lines. So we build everything. We handle all protists, and there is one for viruses, Viper, VIPR. There is one for bacteria, which is called Patrick, PATRIC. And then there is vector-based, that handles the ticks and the mosquitoes, even snails, anything that's a vector. Okay. So those databases are out there. There was no requirement at the time that we were funded, and the same groups have been building these for 15 years to build in the same architecture. That's a double-edged sword. You hate to be mandated to do one thing, so we've each done something different, and I think we've each been successful at certain parts of the problem and the challenges. And we do learn from each other. We get together, we meet, and we talk quite often. What I can tell you we have begun to do is, since we have built the host database, and that is the one thing that all these pathogens have in common, we have now built tools, they're not live yet, but we have built tools to allow people to do a search in their own database when there's a host pathogen data set where they're interested in things that may affect the host, and we can now share gene lists with each other, all right, for the same host genome, and pass those around, and then ask in each of those databases, what can you learn from it? So that has been enabled to facilitate the host pathogen interaction studies. It's not live yet, though. We're still, because it's in alpha phase. It's been going for about 6 months, and we're trying to get the queues going. It's being thwarted a little bit by the fact that we're about up for renewal. We're hoping, anyways. We saw the federal BizOps thing come out a few months ago for small businesses, and ours usually follows a few months after that. And so,
How long are those contracts?
there for 5 years.
5 years?
5 years, yeah. So we're in the 4th year of our 3rd contract. So we're expecting that they intend to continue the program for at least another 5 years.
I wanted to ask you a question also, Jason. So, there's a presidential memorandum now for actually capturing data for an experiment that was initiated by DOE. So what you talked about, the metadata, capturing the entire experiment seems to be ultimately becoming a requirement for actually producing those things. And in fact, I don't know if Ben here from NCBI is here. Hi Ben, I don't know if you could comment on that and how those databases ultimately, how does NCBI deal with all these databases? Is it independent, or do they overlap with some of the things that you guys do in NCBI?
Well, so I think I'm going to speak tomorrow, and I think I'm going to rename my talk a response to Jessica. In terms of dealing with databases, in what sense do you mean?
Well, are those going to be incorporated into NCBI? Are they cross-listed, or are they how are we going to be? How does NCBI deal with those databases as independent entities?
Oh, with other databases,
yes,
like Jessica. So what we're very interested in, and something that we've been talking about quite a bit lately, is getting community annotation into Biostable. The problem is that we can't take everybody's community annotation, because some brilliant grad student could re-annotate everything in NCBI, and there could be a lot of those brilliant graduate students. So actually, one thing that I think is very promising is working with databases like UPathDB as community representatives to get re-annotation of the metadata.
And we do that. So we're the official RefSeq submitter for all the genomes that we have. So.
Right. So that's already starting, but then also to get things ontologically harmonized in terms of raw data sets. I think that's an even larger role that folks like Jessica can play, because actually getting the data out is getting better and better, but having harmonized metadata over a large breadth of data sets is very, very difficult. And so organizations that focus in particular sort of taxonomic areas, I think are valuable in that sense. Did that answer your question?
Yeah, absolutely. And I wanted to also, obviously, you're a computer person, a computational biologist. So to what extent do you think AI solutions will actually play in the long run? Because obviously the databases and integration becomes a very difficult thing. Would machine learning and other, would ultimately be able to resolve or help with implementing some of these data mining?
For mining, yes. I mean, I think they're very good at detecting patterns. They're very good at clustering. They're very good at detecting this thing looks like the others and this thing doesn't. In terms of mining the data, I don't know that they're going to be overly useful in collecting or helping us to sort the metadata. But to the extent that they can determine this thing is not like the other and tell you that your data set might be mislabeled or that this doesn't belong, I see a role for that. What's really needed, I think right now, are very, very easy ways to collect things. And to the extent that inferences can automatically be made, more power to it. You just want to know that they're right. Yeah. All right, let's move on.
Okay, well, thank you very much. Thank you.